Welcome back. In our next key technique, we're going to take a look at a method for determining a protein structure, and that's called X-ray crystallography. So here's the technique. Remember, the function here, the purpose of this is to determine protein structure, because you can't predict protein structure when proteins get bigger than 50 to 100 amino acids or so. So determining the structure of proteins has to be done experimentally, and it's important to understand about protein structure to understand how proteins interact with one another, how they can be possibly targeted by certain drugs, and we also learn about the mechanisms of their actions, how they do what they do, from understanding their structure. So obviously here, if we know about the structure, we can make some inferences about the function. So in X-ray crystallography, crystals of protein can be created and then illuminated using X-rays. So X-rays are shone like a beam of light onto the crystal, and then the diffraction of the X-rays as they strike the atoms of the amino acids making up the protein produces a unique signature, a unique pattern that can be detected and analyzed and used to build a model. So there's a lot of computer simulation and computer modeling going on here. So this is partly kind of some biochemistry in terms of purifying the protein, making the crystals, but then it's a lot of bioinformatics and computational biology to actually do the analysis and build the models. Incidentally, the same approach was used by Rosalind Franklin to deduce the structure of the double helix when she was working with Crick and Watson. There are three stages to the process here. First of all, you've got to make the crystals of the proteins. That's the first thing. Then you have the irradiation using an X-ray source. That's the second step. And then it's the analysis of the diffraction pattern to produce a model of the protein structure. So that first stage, protein crystallization, this is probably the hardest and most time-consuming stage. So the protein has to be purified from a source. Usually it's overexpressed in bacteria, usually E. coli, and then the protein can be purified and concentrated to pretty high concentrations, 25 to 30 milligrams per milliliter. Which doesn't sound like a lot, but it's pretty concentrated for proteins. It's very difficult to get very highly purified proteins, however, and so this is a major technical hurdle. You can't have contaminants because they would interfere with the structural determination in the last stage. The crystals themselves have to be about 20 to 30 microns in diameter and length and width, if you like, and they have to be perfectly formed, so they can't have any inclusions. So that's other matter that's crystallized in with the protein, and they can't have any fractures or anything like that. So they've got to be perfect crystals. So the way that that's done is that you make a small drop of protein solution, and you then expose the protein solution to precipitants. And precipitants are compounds which will bind to water molecules and basically absorb the water molecules out of the solution. So they dehydrate the solution, and as this happens, the protein solution gets more and more and more concentrated until at some point you get microcrystals forming and then nuclear bigger crystals growing. So this takes some time. It's technically challenging, and it requires a lot of skill. So once you've got crystals, then you can put the crystal here in the middle into an X-ray tube and shine these X-rays on the crystal. And as the X-rays hit the crystal, because of the repeating nature of molecules in crystals, the X-rays are diffracted in patterns which are always consistent. Doesn't matter which part of the crystal you illuminate, you'll get the same diffraction pattern. And then you can rotate the crystal and collect multiple data sets, and you end up with a diffraction pattern looking something like this pattern here in the second image here, labeled number 2. Once you've got that data, you have to take each of those dots, the diffraction pattern dots, and you've got to convert them algorithmically into electron density maps. And so that requires some pretty powerful computation. And so then people smarter than me and with a lot more training in this field than me can come up with the positions of certain amino acid R groups and the position of the polypeptide backbone to come up with a model. And so using that model, certain predictions can be made about the positions of certain R groups that haven't necessarily been resolved by the crystallography. And then new crystals can be grown, and the model can be refined. So this takes a long time. Crystallizing and determining protein structure can take a PhD student 4 or 5 years of study. So this is not a trivial task. So that's X-ray crystallography. There are other techniques such as NMR, nuclear magnetic resonance, that can be used to determine protein structure, but this is probably the most easily understood technique. So that's it. And next time we'll come back and we'll take a little look at fatty acids once again. I'll see you soon.